Good evening, everyone. We are happy to welcome you tonight or today, I don't know, depending on the continent you are, for this seminar, web seminar from Calim Institute. We are happy to welcome today Julie Leca. I am co-chairing this lecture with Professor Sylvain Carras in Grenoble. I am Philippe Gollard. I am Professor of Pathology at Henri Mondor University Hospital, University Paris-Est Créteil. And I have, I would say, I was former director of a research group at INSERM with some interest on T-Cell Lymphoma. And I'm happy now to let Sylvain Carras, Dr. Sylvain Carras, introduce himself and introduce Julie Leca.
Good evening, everyone. So I'm Dr. Sylvain Carras from Grenoble University Hospital. I worked formerly under the direction of Laurent Genestier on T-Cell Lymphoma, and it's my very great pleasure to introduce Dr. Julie Leca for his talk. So Dr. Julie Leca completed her master's degree in immunology at the Faculty of Science of Aix-Marseille, and she started her PhD in 2011, at Marseille, in the Dr. Juan Iovanna's team and under the supervision of Dr. Tomasini. During her PhD, the team focused on the study of pancreatic cancer and dialogue between transformed pancreatic cells and stroma. And after her PhD, Dr. Leca joined Professor Takmak at the Princess Margaret Cancer Center in Toronto, a very famous lab in Canada for a 7-year-long post-doctorate position. Dr. Leca's post-doctorate work focused on the different IDH mutations in the context of hematological diseases, such as AITL and AML. And her main subject was the study of the cooperation between TET2 and IDH2 mutation in AITL, which was carried out in collaboration with the team of Philippe Gollard and Professor Luminy in Paris. Since October 2023, Dr. Leca returned to France and joined Dr. Maguire Sata's lab at the CRCL in Lyon, and her team's work is dedicated to exploring the BMP signaling pathway in the biology of stem cells, and again, their interaction with the microenvironment. So, as you know, AITL is a different model than AML, but we are here to hear about ARTL, very specific peripheral T-cell model, and lymphomas that originate from T follicular helper cells, with a prominent role of the tumor microenvironment. And we will hear with pleasure Dr. Leca talk about IDH2 and TET2 co-occurrence and their implication in the tumor regenesis. So thank you very much for all, and Dr. Leca, the stage is yours.
Thank you very much for the kind presentation, and thank you to the Calim team for the invitation. So today I will it's my pleasure to present my data. So I will present you what I did in a Mac lab. So I work on IDH2 in T-Cell Lymphoma. And today I will present you what we did with the first mice model with mutated for IDH2. So just to show you, so the IDH1 mutation was first identified in glioblastoma in 2008. And the year after, IDH1 and IDH2 mutation were identified in AML. And in the same time, another team shows that the 2AG can have some effect in a different enzyme. So our lab published in 2010 the production of 2AG in AML And after in 2002 was a very important year because we discovered the IDH2 mutation in collaboration with Philippe Gollard and François Lemonnier in ITL. But it was also this year that we established the first mice model, the knock-in for IDH1 mutation, and TAC started the Agios company with the IDH inhibitor. So since then, you can see that the IDH inhibitor is, they are very useful in clinical, and there are a lot of different clinical trials. So it was just like to give you an history of the IDH mutation. So the IDH mutation profile differ according to the different disease. You can see that IDH mutation are found in different type of disease, glioma, hematological disease, cholangiocarcinoma, and today I will focus on hematological disease, and specifically the T-Cell Lymphoma called ITL and the AML. So in terms of mutation, we can find different type of mutation. We can find an IDH1 mutation called R132, which is the most frequent one in hematological disease, and two different mutations in IDH2, IDH2 140 and IDH2 172. So in terms of structure, IDH1 and IDH2 are very similar. You can see here, IDH1 is found in a cytosol versus IDH2 in a mitochondria. But what we know about all these different diseases is that when IDH is in a regular context and physiological context, the enzyme converts the isocitrate in alpha-ketoglutarate. And the alpha-ketoglutarate is very important for the metabolism, but it's also a cofactor involving a lot of reaction using the deoxygenase, like the TET2 enzyme, for example. And when IDH is mutated, the enzyme acquires a new activity called neomorphism activity, and there is a production from the alpha-ketoglutarate of the 2-hydroxyglutarate of 2-HG. This oncometabolite is known now to have a lot of different effects in a cell. So the 2-HG can have a lot of impact on the differentiation and also induce aberrant methylation. So the 2-HG can play on a DNA methylation, but also on a histone methylation. And if we focus on a hematological disease, which is very intriguing, is that if we compare the two different types of contexts, so myeloid with the AML disease versus the lymphoid with the ITL disease, you can see that the IDH mutation is very different. So in terms of hotspots, we found the 3 different hotspots in the AML disease, but in a T-cell context, you can see that there is no IDH1 mutation described, only IDH2, and it's most of the time IDH2 R172. So there is a very big difference according to the context, T-cell versus myeloid cell. The other big difference is that there is a very, in terms of commutation, a very different case, because we can see that in AML, IDH1 or IDH2 never co-happen or never co-occur with the TET2 mutation. But here, if we look at the IDH2 patient in an ITL disease, you can see that almost all of them present the TET2 mutation. So according to the context, the commutation happen and are different, and the different hotspots are also very different. So this starts with an idea to explain what's happened and how we can explain the difference of IDH mutation in a myeloid context versus lymphoid context. So this project was funded by the Leukemia and Lymphoma Society, and it was constructed with different teams around this aim to discover the therapeutic implication of altered epigenetic and DMA damage response in hematological disorder. So we have one team, what I work with in a TACMAC in Canada, but we also work with a different team in New York with Ross Levin, Harry Melnick, and Christopher Mason, and of course in France with Philippe Gollard and François Lemonnier. So in terms of classification, I will just give you a quick windows of the PTCL or mature T-cell classification. So it represents 5 to 15% of a non-Hodgkin lymphoma. So it's pretty rare disease, not a lot of patients. It's around 6,000 patients in US per year. And which is important is that the disease happens in a mature T-cell. And you can see that according to the different classification and localization, it's a very heterogeneous group of disease with almost 30 different entities. And today we will focus on one of them, which is the ITL. So before and around like 1974, a lot of paper and different people describe a lymphadenopathy, non-neoplastic, and talk about a hyperimmune reaction. And then in 1980, the clonal nature of the disease was demonstrated, and in 1994, the term of angioimmunoblastic T-cell lymphoma, or ITL, was used. Since then, people discovered that the TFH cells are the neoplastic T-cell present and developing the disease. And in 2011 and 2012, a lot of recurrent and inactivating mutation was described and focused on the epigenetic regulatory gene. So different teams discovered the NMT3 mutation, TET2 and IDH2. 2 years later, the mutation in ROA was described for the first time. And finally, in 2017, the first mouse model described the co-mutation with TET2 and ROA and was very useful for the rest of the story. So if we are looking at the PTCL with the TFH phenotype comprise two different types of disease, the ITL and the PTCL TFH. So today we will talk about the ITL, and it's very important to highlight that IDH2 mutation is only found in an ITL disease compared to the TET2, ROA, or DNMT3, where are found in two different types of disease. So the ITL is a disease, and the neoplastic T cell present a TFH phenotype, so the cells are positive for CXCR13, CXCR5, PD1, and ICOS. It's the second more frequent subtype of mature T cell and represent around like 50 to 20%. And the patient present a poor overall survival. Which is very intriguing and interesting in this disease is that only around 10% of the tumor represents the neoplastic T cell, and up to 90% are composed by the microenvironment. So in a microenvironment, different cell types were described, and specifically vascularization, plasma cell, immunoblast, and follicular dendritic cell. So all of them made like a very inflammatory response and immune dysregulation, and explain why before it was very difficult to classify and identify this type of disease. So normally the regular function of the TFH is to help the B cell response. So in a germinal center reaction, the TFH and the B cell crosstalk via different receptor and ligands, and are very important for the balance and the response of the B cell and for the production of plasma cell. And in the case of an ITL disease, you have a very imbalance of this reaction, with an increase of B cell and B cell proliferation and a plasma cell differentiation as well. And it's important to notice that a lot of patients, and almost all of them, are positive for the Epstein-Barr virus. So the EBV is positive in a B cell, and most of the time, 30% of the patients present monoclonal rearrangement for the IgG. And this can explain why around 10% of the patients develop, in parallel to the ITL, a B cell lymphoma. So in terms of mutation, the landscape of ITL is very unique and very homogeneous. So you can see that around 80% of the patients present the TET2 mutation. So TET2 mutation, it's a loss of function and is leading to the DNA hypermethylation. The second most important mutation is the ROA mutation, which happens in 50 to now 70% of the patients. So what we know about the ROE mutation, it's a dominant negative mutation. And when ROE is mutated, this involves different type of effect in a cell. But in this context of the disease, which is very important, is that ROE seems to be activated of the T-cell signaling. And finally, what we were talking today is the IDH2 mutation, which is found around 20 to 30% of the patients. And this role is very important for the 2AG production, but also for its role on histone methylation and DNA methylation. So in terms of hierarchy, what we know today, thanks to different papers and data published, was that the first hit mutation happened. So it can be TET2 or DNMT3. So in very early progenitor, you have like TET2 or DNMT3 mutation can happen. So you will have your, these two mutations can also happen in a B cell compartment. So this is very important to notice because a lot of patients present the TET2 or DNMT3 mutation in ITL. And then in a very specific TFH population, a second hit happen. So you can have sometime ROE or IDH2 or both of them in the same time. And when the second hit mutation happen, then you have a high proliferation of the TFH. And finally, which is more and more studied today, is the role of the microenvironment. Because we know that the ITL is very depending on the microenvironment, and the microenvironment is very important for the progression of the disease. So it's a very different step in the disease, and all of them are very important for the progression. So it was very difficult for people to work on the ITL disease because there is no cell lines that you can work with, and the PDX model are very difficult to maintain because the importance of the microenvironment. And in 2017, the first very intriguing paper was published and used a T-cell transfer to combine the ROE and TET2 mutation together in a T-cell. So it's important to know that in this model, there is no ITL disease in the mice, but the authors were able to show that when you combine a ROE and TET2 mutation in a T-cell context, you have an implication of the AKT signaling. And then the year after, two different teams show that via different approach. The first one was using the bone marrow transplantation, and they use bone marrow transplant in a TET2 knockout to combine the TET2 knockout and the ROE mutation, and they developed the first ITL disease. And the other paper was published the same year. They use a transgenic mice model this time under the CD4 promotion for the ROE mutation and TET2 in a global knockout. And both of them describe also an involvement of the AKT signaling and the mTOR pathway. So what is known now is that the loss of TET2 is very important to induce the epigenetic background that allows the disease to progress, and the ROE mutation is very important to induce the TCR signaling activation. And both of them, when you combine both of them, the mice were able to develop an ITL-like disease. But now, what do we know about IDH in ITL patients? So IDH was reported for the first time as mutated in 2012. And in this paper, what we know is that there is no difference in terms of clinical survival for the patients between the IDH mutated and the IDH wild-type patients. And in 2015, a paper shows that the IDH2 mutated patient present a very different signature compared to the wild-type patient in ITL disease. So in this paper, they report the same thing, that there is no association with the IDH2 mutation and the patient and the survival of the patient. There is no difference in terms of expression between the two different groups of patients, but they were able to show that the IDH2 mutated patients form a single cluster, very different to the wild-type one. And in this paper, the author also compares the IDH2 R172 mutation found in AML, and this shows that according to the context, once again, if you are looking in an AML versus T-cell lymphoma, the gene and the downregulation an upregulated gene due to the IDH2 R172 mutation is very different. So what they show is that the patient, they compare the IDH and TET2 patient versus the TET2 patient. And they show that when you have the IDH2 mutation combined with the TET2 mutation, the patient presents an increase in terms of gene associated with TFH and a decrease for gene involved in a TH1, TH2, and TH70 gene. And they also show that IDH2 patients present an increase of the VEGF pathway and a decrease of interferon and IL-17 signaling. The author also shows that IDH2 mutated patients present a DNA hypermethylation and an increase of methylation for the H3K27 histone. And they found a very nice correlation between some hypermethylation and gene expression. And they demonstrate that IDH2 mutation is associated with the downregulation of TCR signaling and T-cell differentiation. And then François Lemonnier showed in a study that for the first time, the 2AG was detected in a serum of the patients mutated for IDH. So it's important to notice that the level of 2AG detected in the serum is low compared to the AML patients. And he was also able to demonstrate in a cell, a mutated cell, that 2AG is produced by at the intracellular level. And then by immunochemistry and immunofluorescence using IDH2 R172 antibody form with a co-staining with different marker for different type of cell, it shows that the IDH2 R172 mutation is found in a CD3 cell and specifically in a ICOS positive cell. And demonstrating that, for example, the CD8 population, in a CD8 population, the IDH2 172 mutation never co-happen, neither for the macrophages or the B cell. So this confirmed for the first time in a patient that was thinking about the IDH2 mutation that it's very restricted in a TFH compartment and it's not found like TET2 or DNMT3 in other cell type. So with all this rationale, the specific distribution of IDH in ITL, but also the fact that IDH co-happen with a TET2 mutation, which is very different with people described for AML, the specific signature of the patient, and the fact that there is no mice model available, we decide to work on IDH2 mutation in this disease. So François Lemonnier, for the first time, tried to establish a mice model using the VAV-3. So according to the different VAVs that you can use in a mice, you can induce the mutation at different moments and different time in a hematopoiesis. So you can see here, like with the VAV3, you induce a mutation in hematopoietic stem cell compartments. So François did the knock-in mice and studied the 3 different hotspots for the 3 different IDH mutation using the VAV3. So once again, the IDH mutation will be present in all the different compartments and all the different cell types. And it was very important because compared to the overexpression that people published before, it was more like a physiological level of IDH expression, because it's important to understand that the IDH mutation is heterozygote, so you keep one wild-type allele, and the balance between the mutated and the wild-type form is very important for the activity of the enzyme. And what he shows is that the level of 2AG is different according to the different mutation. So here, coming from the mice, he isolates 3 different populations, the myeloid cell, the T cell, and the B cell. And here you can see that even if there is some difference, because the red one here, which represents the IDH2 mutation, all of them, the different hotspots are able to produce the 2AG in a myeloid context. But if you are looking what's happening in a T cell context, you can see that there is no 2AG produced by the IDH1 mutation. And if you are looking the IDH2 mutation, only the R172 was able to produce enough 2AG in a T cell context. So this can explain why it never happened to have some IDH1 mutation in ITL context, but it's mainly IDH2 172. And because the 2AG is known to have some effect on a TET2 enzyme, so normally the TET2 enzyme converts the 5MC in 5HMC. So if you have some 2AG production by the IDH enzyme, you inhibit the TET2 enzyme and you have a decrease of 5HMC. And here, if you isolate the T cell, you can see that the only condition which we can observe the 5HMC lost is the IDH1 72. So the same that produced 2AG is also able to inhibit the TET2 enzyme. And after, the question was, can we explain this difference of 2AG production by the different expression of the IDH1 or IDH2? And what François showed by QRT-PCR here or here by a fax is that the IDH1 is very expressed at the very low level in CD3 compared to the myeloid compartment. So in terms of expression, this can explain why the IDH1 mutation never happened in a ITL patient. And if you are looking for the IDH2, there is no significant difference between the IDH2 in a different cell type. So then François start to analyze the mice to see what effects can be induced by this 2AG production and inhibition of TET2. And he found that in R172K mice, there is an impact of the thymic development. There is a decrease of the thymocytes and also an increase of the double negative population correlated with an increase of the double negative and a decrease of double positive cell. So the thymus was impacted by the mutation. And if the number of CD3 was the same among the different mice, it shows that there is an important decrease of naive T cell, CD4 T cell in 170K mice, correlated with an increase of the activated marker for the CD4. But when he was looking for the TFH marker, CXCR5 and PD1, there is no difference between the three different mutations. So when using the VAV3 mutation, this study was allow us to say and to understand why probably the IDH1 mutation never happened in a T-cell lymphoma. And it's mainly IDH2 R172 due to this high ability to produce a 2AG. So he was able to detect the IDH2 mutation induce the 2AG production, induce and inhibit the TET2 enzyme via the decrease of 5HMC. And this mutation was able to affect the lymphoid development, but the mice never developed any T-cell disease. So then we decide to change the CRE activity using the CD4 CRE activity. So the CD4 happened here, so at a later time compared to the VAV CRE, and you will have the mutation in a T-cell compartment. So both of them, CD4 and CD8. And this time, we decide to compare the IDH1 versus IDH2 versus the double mutants. What we show using this transgenic mice model is that this time, we did not observe any impact on the thymus development. So there is no immature impact as we have before using the VAV3. We also show that the T cells are able to produce a 2AG. And here is very important to notice that in orange, you have the IDH2 single mutant versus the red, which combine IDH2 and TET2 mutation. And you can see that the level of 2AG is higher when you combine IDH2 and TET2 mutation together. And very early in the mice, you can see in red that we were able to detect an increase of the TFH population, stained here by CXCR5 and PD1 marker, associated with an increase of B-germinal center cell. So this is very important because as I told you before, the ITL disease is not only a TFH disease, it's very like a B-germinal center disease. So it was very encouraging to see some effects in a B-cell population, and specifically in a B-germinal center population. But unfortunately, we were also able to detect high amount of immature myeloid cell in this mice, specifically after 8 months. And when we isolate this cell and dosage the 2Ag, we were also able to detect a very high production of 2Ag in a myeloid compartment. So to understand what's happened in our model, we decided to cross the CD4 with the tomato mice to see when the tomato can be detected. And we were able to see that the LSK population expressed the tomato around 10%. So that means that very early, some progenitors were able to express the tomato. So in our case, that can explain why the IDH mutation can be found in a myeloid compartment. And because the expression of IDH2 is very high and the ability of IDH2-172 is very high to produce 2Ag in a myeloid context, it can explain why these mice produce a very high amount of 2Ag. It was not due to the T-cell, but due to the myeloid cell. And finally, we were not able to detect any clonal T-cell rearrangement in these mice. So when we combine the two mutations, IDH and T2, using the CD4 tree, we were able to observe an increase of 2Ag production, an increase of TFH population, and an increase of the germinal center B cell. But unfortunately, the mice develop an immature myeloid neoplasm before to develop any T-cell lymphoma. So to avoid this issue with our model, what we decide to do is to use a T-cell transfer. So we use our transgenic mice at very young age, around 2 months. We isolate the T-cell and we characterize the CD3 population. So you can see that at this time, if we are looking at the CD4 population, there is no difference between the different mice. All the mice have approximately the same amount of CD4 cell. And in terms of TCR repertoire, you can see that it's very homogeneous according to the different group. The cells were well able to produce the 2Ag, and once again, when you combine the two mutations, the 2Ag was increased. So once we isolate and characterize our population, we decide to transplant them in a TCR knockout mice. So the TCR knockout mice are like a mice without any T cell. So we can after follow and study the T cells what we inject. And because it was a very difficult model to work with, we decided also to help the system a little bit. So we stimulate the mice with a cheap red blood cell. So this induces an activation of the B germinal center. It's known and it's used by different groups for different ATL model because it brings some immunization to your mice and it increases the TFH and the B germinal center reaction. And then we study what's happening in our mice. So we found that there is no change in terms of global CD3 population, even if it was very heterogeneous according to the different mice. At the median level, it's very similar. But you can see that in a double mutant, when the TCR was injected with a double mutant cell, we have an increase of the CD4 and CD8 ratio. So here I present you all the data. So the black dot was a mice injected with a cheap red blood cell, and the gray dot was a mice injected with a PBS as a control. So only when you combine the two mutations together, you have an increase of the CD4 population. And for the first time, we were able to detect in this condition a monoclonal T-cell disease. Then we decide to characterize the T-cell, and we show that when we combine the two mutations together, we have an increase of the TFH marker. Once again, we use ICOS and PD-1. And here you can also see that when the mice were injected with a TET2 mutated T-cell, they also present an increase of the TFH cell. But this was less compared to the double mutant cell. And then we analyze in terms of histology what's happened in our mice and in our tumor. So the mice develop a splenomegaly as well as tumor mainly in a mesenteric lymph node. And we were able, by immunochemistry and different immunostaining, to see that the spleen and the lymph node present a destructive architecture of their regular architecture. And we were able to detect the CD4 T cell, but also a very important population of a B220 B cell, as well as the plasma cell, and also vessels that we can see here by the CD31 staining. And we were also able to detect an increase of B germinal center reaction in a double mutant T cell, which was very important for us to validate our model, because to be classified as ITL disease in a mouse, you need to show that you have your impact of the T cell, which we have with a clonal repertoire, but we also have the TFH marker, but we also have a very important impact on the microenvironment. So this was to show you what's happened when we combine and analyze a very small percentage of all our cohorts. We show that when you combine the two mutations together, IDH and TET2, injected in a TCR knockout, the mice develop, around 60% of the mice develop an ITL-like disease. And you can see that between the stimulation with or without chip red blood cells, there is not a lot of difference. However, if we study what's happened in the case of you inject the TET2 mutated cells, it's very different. So around 20% of the mice develop an ITL-like disease under chip red blood cell stimulation, but most of them develop a B-cell lymphoma. And it's very different because the chip red blood cell stimulation in a TET2 context alone seems very important compared to the PBS injected one. So then we try to understand what's happened in a B-cell compartment. What we found, and if we compare the TET2 versus the double mutant cell, we found a decrease of the naive T cell in both of mice compared to the IDH alone or the wild type alone. But we were able to detect an increase of the plasma cell only when we combine the two mutations, which correlate with an increase of IgG production in the serum of the mice. And then, because we know that the crosstalk between the TFH and the B cell are very important, we study in a TFH cell as well in a B cell the different marker and ligand. So, for example, in a TFH cell, we compare the expression of ICOS, PD-1, CD40 ligand, and CD28, and the correlation with the B cell compartment was ICOS ligand, PDL1 ligand, CD40, and CD86. And you can see that each time, it seems that the TET2 and the IDH double mutant cell seems to have the same direction, but it's increased a little bit more when you study the double mutant cell. So in both of the case, TET2 and double mutant, we were able to detect an increase of ICOS and PD-1 in a TFH compartment, but also on the B cell. So that means when we inject in your mice, which is like totally regular TCR knockout mice, you inject some mutated T cell, we were able to detect a very important impact on the B cell population, characterized by an increase of CD40 and CD86. So it was very important to notice that nothing happened with the IDH1 mutation alone, but when we inject the TET2 mutated cell, we have a very important impact on the B cell. So this can explain why we have a frequent clonal B cell proliferation. And as I showed you before, around 40% of the mice treated with a chip red blood cell develop a B cell lymphoma. In the opposite, when you co-inject the IDH2 and the TET2 mutated cell, we found an increase of B germinal center action, more characterized by an increase of B germinal center, also an increase of plasma cell, and you have a development of the IETL disease, and in parallel sometimes, probably a B cell proliferation. So to understand what's happened and how we can study the T and the TFH and the B cell crosstalk, we try to do some in vitro assay. So what we did, we isolate the CD4 naive T cell with a different mutation, and we use a specific media to induce a TFH population. And what we show is that this time, you can see that when you study the IDH mutation alone, or the double mutant, you have an increase of the ICOS expression as well as PD-1 expression. But when you compare the double mutant versus the IDH alone, you can see that the effect is better when you combine both of the mutation compared to the IDH2 mutation. And it was very intriguing to see that in the same time that you increase the TFH marker, we can also detect a decrease of the CD4 expression. So when you put the cell in a media specifically to induce the TFH differentiation, we were able to see that the double mutants present an increase of ICOS and PD-1, but a decrease of CD4. So then to understand what that means and how this can impact the B cell, we decide to realize some co-culture. So we isolate the B cell from a wild-type mice, and we use the same B cell in all our different conditions. And we put with this regular B cell some T cell or TFH cells that we isolate from the mice with a different mutation. And here what you can see is that if we are looking in this co-culture, specifically the B cell, we see an increase in terms of number and percentage when the B cell are co-cultivated with IDH2 mutated T cell or IDH2 mutated T cell. So the IDH mutation seems to have a very important impact for the B cell proliferation and number. So to understand how we can explain this impact on the B cell, we decide to characterize the T cell by transcriptomic and epigenetic. So what we did, we isolate the CD4 T cell and we see what's happened in RNA-seq. So here it's a wild-type mice, the IDH2 mice, the double mutant and the TET2 compared to the wild-type mice. And what we can see is that there is not a lot of downregulation or upregulation in a single mutant, IDH or TET2, and there is not a lot of common gene between them. But you can see that when you combine the two mutations together, you have like a very and completely different pattern, completely different with the single mutation, IDH or TET2. So when you combine the two mutations together in a T-cell context, something happens, and it's very important for the T-cell in terms of gene expression. We can confirm what we showed before, so we have a decrease in terms of CD4 at the RNA level. So that can explain why we were able to observe a decrease of the CD4 by FACS. And we were able to, when we did like a GSCA, to see that the modulation of the gene were mainly associated with immune response, chemotaxis, and metastasis. And here I plot the most important cytokines that we observe according to the different mutation. And we found some important cytokine involved with the germinal center reaction that you can see here with the GC letter here, so IL-22, CCL20, for example, OX40 ligands. And we also find other cytokines involved in the angiogenesis pathway, and I plot them with a red arrow here. So in our model, when we decide to investigate in terms of gene signature what's happened when you combine the IDH and TET2 mutation together, we found that a lot of cytokines can explain the effects that we were able to observe in the mice in a microenvironment, specifically in a B cell, but also in a angiogenesis. And because we know that IDH2 and TET2 mutation are important targets in epigenetic, we decide to realize some ATAC-seq to determine the chromatin structure of our cell. So the same sample were used for RNA-seq and ATAC-seq, and you can see that once again, we have a very and completely different signature in terms of chromatin conformation when we compare the different mice. And when you combine the two mutations together, you have mainly a closing chromatin, which correlates with the downregulation of the gene, and very few modification in an IDH or TET2 single mutant. We observe a very good correlation if we compare our data from the ATAC and the gene expression, meaning that the downregulated gene were mainly associated with closed chromatin area, and upregulated gene was more associated with open chromatin area. And because we know that TET2 is very important for the DNA methylation and IDH can be important for the histone and DNA methylation as well, we decide to rely some bisulfite sequencing on this TFH to study the DNA methylation. So we also observe a very high mark of hypermethylation in a double mutant, and the pattern was once again very different if we compare the double mutant and the single mutant. But this time we were very surprised to see that the methylation correlates with the differential, with the gene expression only for few genes. So here it's all the genes that we found modulated in our cells. So we have a high hypermethylation correlated with the downregulation of the gene, but it only affects a very small number of the gene. So we can explain why the CD4 is downregulated in the gene. It's because the CD4 promoter is hypermethylated in the double mutant cell. And in fact, we found a lot of TCR signaling gene downregulated and hypermethylated in our double mutant cell. But this also means that other process, like for example, the histone methylation, can explain the difference in terms of gene expression, because clearly the DNA methylation was not enough to explain all the downregulated and upregulated difference that we saw. And one of the gene was very important for us because we detect in a double mutant a decrease of Fas ligand. And it was also very intriguing to see that we have a very good correlation between the Fas ligand expression and CD4. So normally, some paper published before that if you have a very good TFH activity and reaction, you have an increase of the CD4 expression. And in our case, we can see that in a double mutant, in a red dot, we have a very good correlation between CD4 and Fas ligand, but in terms of downregulation. That means that our cell downregulates the CD4 expression as well as the Fas ligand. And Fas ligand is very important in a TFH to induce the death of the B cell. So it's very like an important balance to control the B cell fate. And we confirm this data because it was RNA sequencing by fax in our mice model. And we observe that in the IDH and double mutant mice, we can see a downregulation of the Fas ligand expression. So because of the important role of Fas ligand in a B cell, we decide to use again our co-culture model. So once again, we isolate the B cell from a regular mice, wild-type B cell, and we put the B cell with a different TFH with a different population. And what we can see is that when you co-cultivate the B cell and the TFH, mutated for IDH or double mutant, we have an increase of the viability of the B cell. So the question was, what's happened when you put a Fas antibody which is an agonist and which mimics the Fas ligand action? So we did the experiment, and in fact, we have a decrease of the viability of the B cell in all the conditions. So that means that the B cell were able to reply to the Fas signaling, but something else is impacted. And because we have the downregulation of the Fas ligand by RNA and by protein level, what we did is that we isolate the B cell, from our mice, from our sick mice, and we put this B cell with a Fas ligand protein. So it's a recombinant and artificial model. We just add the Fas ligand in our media. And this time, when we add the Fas ligand, we were able to detect an increase of the apoptosis of the B cell, which correlates with a decrease of their viability. So what's happened in our model, at least one mechanism that we were able to identify thanks to our RNA sequencing, is that in a TFH, you have a downregulation of the Fas ligand. So that can explain, at least in part, why the B cell continue to proliferate, because they were not able to induce the apoptosis. And finally, we want to know if our mice model was relevant or not for the disease and to study what's happened. So we used some RNA sequencing. We did some RNA sequencing from our tumor, and we were able to have a good match with some gene signature found in ATL patients mutated for IDH2. And because our data also points the fact that IDH2 seems to be relative to the plasma cell and angiogenesis, we use immunochemistry. And we found that using immunochemistry, we have an increase for a certain plasma cell marker in an IDH mutated patient compared to the wild-type ATL patient. And using the RNA-seq data on a patient sample, we show that high IDH2 VAV correlates with more plasma cell. So it seems that even in a patient, the effect that we observe in our mice with the increase of the plasma cell is also true in a patient mutated for IDH. We also found in our mice an increase in terms of gene signature associated with the angiogenesis. And in fact, when we did some staining in immunochemistry on the ATL mutated patients, we also find an increase of a marker for angiogenesis. So it seems that thanks to our mice model, we find some interesting correlation in the IDH2 mutated patient. So our mice model seems to mimic, at least for some parts, what's happened in the IDH2 ITL patient. So here it's a summary to what was published before and what we did. So before, most of the paper was associated the TET2 and the ROE mutation together. And I think it was a very good model to start with because you really need to have the very specific mutation to understand what's happened in terms of epigenetic, but also in terms of TCR activation. And this was a very good combination, the TET2 and ROE. And clearly the different paper always show some difference happen, of course, but it was very strong to see that when you combine the two mutations together, you have an increase of the TFH program, TFH proliferation, and TFH modulation. And the author was also able to describe some B-cell proliferation and differentiation in plasma cell. What we did in our case, we show that when we combine IDH2 and TET2 mutation together, we have an important reprogramming in terms of signature, genetic signature due to epigenetic impact. And among the different effects, we have an effect on the angiogenesis. We have an important effect on the TFH and the B-cell crosstalk, as we show by the fast ligand. So we have a very important effect on the microenvironment. We have an increase in B-cell population due to the increase of the survival and their increase in the plasma cell. And we found a downregulation of the TCR signaling, but we cannot find a very clear effect on the two mutations together in a TFH cell by themselves. So clearly, the ROE mutation is missing in our model. This is like a negative impact of our study, is that I think it's very important to understand that the TET2 mutation, but also the ROE mutation are the two important points for the ITL disease, and we were not able to add the ROE mutation. So our model was very important to explain and to understand what's happened when you combine the two mutations together, but clearly the ROE mutation is missing. And this is also why we use the cheap red blood cell stimulation to increase and to try to compensate this loss of TCR signaling. But I would like to finish talking about a few perspectives, and specifically the TFH and the B-cell crosstalk. So different papers show that more and more paper about ITL shows that the B-cell are really important for the disease. So a paper was published like in 2022 by a Japanese group, and they show that the T-cell, and specifically the TET2 mutated T-cell, B-cells are very important for the niche of the ITL. And it's very different in their model if you compare the B-cell mutated or not for the TET2 mutation in terms of disease progression. Another group also shows that if you stop the crosstalk using a downregulation, if you stop the crosstalk with a T and B-cell and TFH cell, you decrease the size of the tumor. But finally, it was also, so more and more studies show the impact of the B-cell, but finally, if we are looking at the clinical data and what's happened with the clinical trial, when people use the rituximab, which is a CD20 antibody, so when people use and target the B-cell compartment, there is no effect for patients in terms of progression. So there is no effect, no benefit effect in terms of survival for this patient. However, I think that the B-cell is very important to consider in ITL disease for different points. So most and more people and talk about the EBV context. In fact, 66 to 86% of the patients present an infection of EBV. And more people also talk that the TET2, TNMT3, and now NOTCH1 mutation found in a B-cell. And in all the papers that you can read, it's mainly EBV or genetic anomaly that people are thinking about driver of B-cell proliferation. But clearly in our mice model, we were able to show that we were not infected by the EBV, and the B-cell at the beginning have no genetic abnormality, but just put them in contact with neoplastic TFH was enough for them to drive a kind of proliferation and develop a B-cell lymphoma. And it's also very intriguing to see that up to 30% of the patients develop a monoclonal or oligoclonal rearrangement for the immunoglobulin gene, and 10% of the ATL patients can develop in parallel a DLBCL, which is a type of B-cell lymphoma. So study the correlation and the crosstalk of TFH and B-cell, which I think it's very important for the ATL disease, but not only for the ATL disease. Because if you are looking the opposite, if you are looking on the B-cell side, the B-cell, some of the B-cell, like for example, the follicular lymphoma or DLBCL are also a disease which impacts the B-germinal center reaction. So finally, if you are looking on the TFH or on the B-cell, maybe we can be able to understand more what's happened. And it's very interesting to see that in de novo DLBCL, when you compare the percentage of TET2 and DNM3 mutation in EBV negative or positive context, you can see that it seems that EBV also play an important role in a B-cell lymphoma. So I think the story between the TFH and the B-cell can be very intriguing for the science in general, not just for the ATL, but for the ATL, for the B-cell disease, but also for all the people working on the B-germinal center reaction. And finally, I would like to finish this talk by showing you this idea that we developed with Tax, thanks to a very talented illustrator, Sylvain Bagny, that helped you to modelize our idea. We are thinking that the ATL is way more than just T follicular helper disease. And using mouse model and human sample, we need to investigate how the TFH neoplastic cells, represented here as a devil, affect the microenvironment. We show in our study that the mutation can impact the physical and direct link between the microenvironment and the tumoral cell, which is represented here by the checking hand. But we also know that other type of signal, like cytokine or secreted factor, which are represented here by phone and antenna, are also very important to modulate the microenvironment. And we are thinking that these TFH cells by themselves have the mutation, but they affect the important effect to study is really the microenvironment to understand what's happened and how the disease progress. So finally, I would like to thank everything from the TACMAC lab because it was a very long postdoc and a lot of people were involved in this study. I have a lot of help for the animal colony because it was a lot of work, and I'm very grateful for all the help that I have in a lab. And we have a chance to develop this project in collaboration with a very talented group in New York. With our collaborator, we help us to characterize the epigenetic and transcriptomic assay, Harry Melnick and Christopher Mason. And of course, with François Lemonnier and Philippe Gollard, because even if we we try to understand what's happened in terms of histology in our mice, it was very difficult. And I think if you want to work on the lymphoma, because it's so complicated and the classification is very difficult, you really need to work with people who know what to look. And Philippe and François help us a lot to develop this project. So finally, I would like to thank everyone for your attention and the Cali team for the invitation. Thank you.
Thank you very much, Dr. Julie Leca, for this very comprehensive lecture, which opened new concept about the role of these two mutations, TET2 and IDH2, to synergize to induce modification of the tumor microenvironment. And I think this is a nice study where you show finally how the mouse model recapitulates, at least in part, what we see in the pathology of the human disease, in which IDH2, for example, is not found in the TFH lymphoma, which are not angioblastic T-cell lymphoma, but only in angioblastic T-cell lymphoma, where you have a lot of microenvironment, including plasma cells and vessels. So now this lecture is open for discussion and question. Maybe Sylvain, you want to start?
Yeah, I didn't see any question in the chat, so I may begin. Thank you very much. It was very interesting and inspiring in terms of fundamental and translational science and collaboration between different teams. And I have some question, maybe not in the heart of your project, but that questioned us a lot in the team in Lyon. And the first question is the role of the TCR stimulation in your model. Do you think it's only a hit to overcome the downregulation of the CD3, the TCR pathway, or you think that there is a specific role of the TCR signaling in the lymphoma genesis steps?
So at the beginning, yes, we are thinking that the chip red blood cell stimulation was really needed for double mutant cell to develop the disease. But finally, when we combine the IDH2 and TET2 mutation, what we show is that there is not a difference in terms of disease, percentage of disease in a mice, but the mice injected with a chip red blood cell present, for example, a loss of CD3 in the membrane, which is found in a patient, and presents also more B-germinal center percentage. So everything seems like better, but it's not like mandatory for the mice to develop an ITL disease.
Okay, thank you. Maybe you investigated the role of fast ligand signaling on the B cell viability, but we know that fast ligand is implicated in the AICD. Did you look at the role of fast ligand to modify the viability of the T cell in your model?
No, we didn't look. So at the beginning, so we know that fast ligand is also very toxic in terms of liver toxicity. So that's why it was interesting to see that we have a very nice correlation between our mRNA sequencing and what we observe in our mice and in vitro. But for us, it was not a very good option in terms of therapeutic.
I think there's a question from the chat from Bertrand Adel, who thanks you very much for this very complete story. But can you elaborate, Julie, on the role of clonal hematopoiesis in disease development? You know, clonal hematopoiesis is frequent in this disease. So can you elaborate on this?
So yeah, we know that like 60 to 70% of the patients present a clonal hematopoiesis. So I think it's very important to remember that TET2 and DNMT3 happen first, and then IDH2 mutation. But what IDH2 mutation is doing in terms of clonal hematopoiesis, it's not clear for now. And specifically because it's not found in other PTCL, it's very... I think the second is like ROE and IDH2 mutation, which happened later in a T cell. I don't know if like can elaborate a real role of clonal hematopoiesis or if just happen in this specific T cell population because of some context. I don't know, like that for IDH2, I don't know what to say about the clonal hematopoiesis.
But maybe we could elaborate about the role of TET2 mutation in the B cell compartment. And you already answered about the role of clonal hematopoiesis through the B cell compartment. I understand, as I understand your talk.
Yeah, so we know that the TET2 seems very important. It was two very nice studies. One coming from Japanese, one coming from German people, shows that the microdissected B cell really presents the TET2 mutation as well as the DNMT3. So it was very difficult at the beginning because to investigate and understand what's happened, it was difficult in terms of sequencing technique, in terms of resolution. But now I think it's way better compared to before. We start to understand exactly what's happened. And the TET2 mutation and DNMT3 for sure seems very important for the development of the disease and also to affect the microenvironment. But I think it just like there is a beginning, because even if this is for so long, before the new technique and the energy development, it was very difficult to understand what's happened. Now we start to understand better, for example, the role of the B cell, but what's happened for example, for the follicular dendritic cell. Nobody really know what's happened. And they probably are really important as well.
So if we go back to the model, you say that probably the stimulation by sheep red blood cells is kind of a surrogate of ROI mutation. Is it something which is really? I mean, this is an hypothesis, of course, due to the role of ROI. But what do you expect of the triple mutated mice? I mean, ROI, TET2, and IDH2. This is my first question. And the second question will be, because you show nicely how the crosstalk between the TFH cells and the B cells is so important. So what would you recommend as a therapy or as a perspective for developing a new therapy in this disease to better treat the patients?
So for the first question, I think at the beginning, as you said, it was an hypothesis, the sheep red blood cell, because, for example, it's also used in other mice model when they have the ROE mutation. For example, the Cortes paper also use the sheep red blood cell every 3 weeks. So I think it's something to increase the stimulation. And because it's not so, I mean, it's very long disease in a mice, so I think you help the system a little bit. But clearly, we show in our model that it's not necessary for the development of the disease. However, I think that the ROE mutation and everything what's happened due to this mutation in terms of TCR signalization is really important, and it's still missing in our model. So we have some idea and some hypotheses to what's happened for the TFH and the B cell crosstalk, but clearly we cannot really study what's happened in a T cell compartment because one of the main mutations is missing. So in terms of therapeutic, I think both of the aspects need to be considered, the epigenetic, but also the TCR signaling. And I think it's very like the two different aspects that we need to remember when we are thinking about the ITL disease is that you have epigenetic effects by IDH2, TET2 mutation, DNMT3, which is found in all the mice because TET2 and DNMT3 are very necessary and found in, I mean, TET2 in almost all the 90% of the patient now, according to the number, but the ROE mutation is also very important. So I will probably start with a combination of both, like epigenetic drug and TCR signaling drug.
We have another question for Bertrand Nadel, and it was I had the same question. In your model, what role do you can infer for AKT-mTOR pathway and notably under the ICOS signaling? You show an upregulation of ICOS, particularly in the double mutant, but not the expression of ICOS ligand in the B cell. Do you think there is another compartment of cells that express the ligand? Do you think that there is a role for this crosstalk in the cell, the tumoral cell survival?
Yeah, so you're right, we didn't check the ICOS ligand. We know that the ICOS ligand can be expressed by the B cell, but it can be also expressed by, for example, the follicular dendritic cell. So it can be different type of cell in the microenvironment. We have a very high increase of ICOS in our model, but we never really looked at ICOS signaling. And after, I think, and maybe I'm wrong, but I think that the PA3 kinase, it's mainly found in the wild-type patients, not in the IDH2 mutated patients.
Okay, but it was an interesting question for a translational point of view, as we know that we have ICOS antibodies, anti-ICOS antibodies that are under current development, and it may be another model to address the question as the paper of Cortes did in 2018, I think. I don't see any other question, but I have one more, if you allow me. Did you look at the histone mark and the H3 key 27 3-methyl mark in the model? Do you think that it cooperates with the DNA methylation to the phenotype of the tumor cells?
So it's a very good question. So for technical reason, when we start the project, we really want to investigate the transcriptomic and the epigenetic on the same sample, because it's so difficult, if not, to compare what's happened, specifically in terms of epigenetic marks. You can have like so many different factors who can influence everything. So we really want to study everything on the same pool of cells. So the ATAC-seq and the RNA-seq, it's okay in terms of material. But for the histone application, you need to have like a lot of material for this type of technique. And because we were also able to collaborate with Harry Melnick and Christopher Mason in New York to develop a very nice pipeline to study the DNA methylation, we start with the DNA methylation. But we were pretty like, unfortunately, we cannot see a very good correlation. We found some interesting gene. Clearly, we show like for example, the CD4 is downregulated due to the hypermethylation on its promoter. But in terms of the number of that we can associate with the hypermethylation and the gene expression was very low. So I think it's a very good point, and I think it's something that we need to do. But in terms of technique, it's not, I mean, it's complicated.
Thank you very much. I think I don't see other questions from the chat. Do you see, Sylvain? No.
No, I don't see any other question. And maybe I have one last question, but it's more about the role of TET2 and maybe the NMT3A loss of function mutation on the as a permissive state for the EBV infection. And do you think that, I didn't get maybe in your presentation, if there is a temporal sequence, temporal steps, with first a T-cell proliferation and lately a B-cell proliferation, if you are able to answer to a co-occurrence of the two phenomenon in the B-cell and the T-cell compartment. I don't know, maybe it's two questions. Sorry.
So first, in my mice model, at the very beginning, we study what's happened in very young mice, like around 3 to 4 months after the transplantation. And already at this stage, we have a very nice increase of the B germinal center percentage. But we never look in terms of clonality of the B cell. We checked the clonality of the B cell later when the mice were pretty sick, and we were able to see like a clonal B cell rearrangement, but we never checked before. So my guess is that because we induce the mutation in a T cell, we have first a T cell impact and later a B cell impact, but I'm not sure it's what's happened in a patient. And I'm not sure it's totally true in my mice neither, because finally, when we also isolate the B cell to know what we can find in terms of mutation, and we found some mutation in the B cell. So clearly the B cell was impacted, and we know that it's not impacting from the beginning because due to our mice model and the way that the mice model where we did our experiments. But clearly the TH mutation was able to induce by some signal or maybe the role of the microenvironment or other cell, we don't know exactly what's happened, an impact on the B cell. And for the patients, for the EBV, I don't know exactly, and it's very also difficult to find some correlation because most of the time it's very difficult to associate the EBV status with the mutation, for example. And also when the patients are described for the ITL disease, sometimes you have B-cell proliferation, but not all the time monoclonal or oligoclonal. And I think it's one of the things that it's like missing, if I can say, because for me, for example, to understand what's happened in my mice, when I look the description of the patients, it was very difficult because now we don't know if it's due to EBV, to some genetic alteration in a B cell. And we don't know, for example, if the 10% of the ATL patients will develop T-Cell Lymphoma, what type of mutation this patient presents.
I may have another additional question, which is a very limited question from pathologist. In the human disease, the IDH2 mutation is associated with cells, TFH neoplastic cells that have a large clear cytoplasm. So I wanted to know, we don't know exactly why so far. I mean, it has not been clearly investigated, but several groups found the same thing. And we can recognize easily now on the microscope the cases that are IDH2 mutated. And I wanted to know whether you had maybe an explanation from your mouse model or not for this observation.
So we were not able in our mice model to see a very nice increase that you can see in a patient. We saw some of the cells with like an increased size, but it was not as clear as that you can find in a patient. And in terms of explanation, what happened in this cell? No, I don't have any real idea.
I think it could be mitochondria or I don't know what, but okay. Maybe it's yes, Sylvain. You have still a question?
No, it's not a question, but I think it's more a perspective or a discussion. You did not explain a lot the reliability of your model for translational development and drug targeting. But I saw in the beginning of the talk that there was some people from industry, and I think we have to emphasize the interest of this type of model to develop some strategies and new inhibitors, and I think that it's a good thing to highlight at the end of the discussion, if you allow me to do it.
During that time, there has been one additional question on the chat from Dr. Gamze Tari. Hello, thank you so much for your interesting talk. I am having difficulties to engraft IDH2 mutated ATL tumor into immunodeficient mice, while there are AML and MDS PDX models developed with IDH2 mutated samples. What can be the reason of this failure in ATL PDX? That's a good question.
So I will start first with the Sylvain question about the therapeutic. So what we show in our model, because of course the disease happen in like 10 or 11 months, so you cannot use the mice. But what we show is that half of them are transplantable in NSG mice. So this is like a good start. And if I did not use anything, it's clearly because I have no time enough to do any drug testing or everything. I did it on a cell, but not on the mice. After the NSG is not the best, we also try to engraft the cell in a TCR, in a B6 mice, for example, to have the role of the immune system, but it was not very successful at all. And most of the time, the issue that we have is that when we try to transplant our model, we have a B-cell lymphoma. So it's, I think it's very interesting to do it. And of course, it's, that's the aim of, the goal of everyone to understand what's happened and how it can be used for patients. But right now, our mice model is not perfect. We can use it in an NSG mice, and we can maybe start with some antibody, inhibiting antibody, but we need to be careful of the NSG model. And we need to keep in mind that it's not the best strategy. And for the engraftment and the difference between the ATL and the AML, I think one of the, just my point of view, but I think in ATL, the difference is the microenvironment. Because we know that the percentage of cells are very low, 5 to 10%. So clearly there is a need. The cell needs to have the microenvironmental rod to grow and to survive. Yeah.
So one key could be, I know it's very difficult model, but one key would be humanized model with fresh PBMC that could allow a more prominent microenvironment in these mice.
Yeah, I think this is one of the options that we can. I mean, we need to see, but to find. I think it's very difficult to find some good response because first we need to have the good mutation. I think this is really important. More and more mice, there is like different mice model using mutations that we cannot find on the patients. And I think when you are talking about the ATL and specifically the epigenetic marks, this is really important to understand what's happened in mice, because even if you don't know what's happened in mice, it never really work all the time, as you can say in human and in patients. But we need to have a mice model as not the best, but as close as we can from the reality and the relevance of the.
I think we are getting late now. It's dinner time now in France, at least. We must, with Sylvain, thank everyone for attending this web seminar. And we thank you again very much, Julie. And we, of course, all the attendants are invited to join also the next Redtalks of the Calim Institute. And we'll be happy to meet you at this occasion. So thank you again, Julie, and everyone for attending this lecture. Thank you all. Bye-bye.
Thank you very much.
Bye-bye.